Good evening, everybody. Wow, I'm surprised. Like 50 people of you have dialed in so far, and I hope very much will not disappoint you. As you probably know, my name is Alfons Weber, and I would like to welcome you to the first ever Atomly live stream. So these are, of course, very special times, and we obviously couldn't meet in person during this pandemic outbreak. But obviously, we want to protect not only ourselves, but also the people we love. And so in these difficult times, our usual ways of living have been somewhat turned upside down. At least they have substantially changed. And who knows what will actually happen in the future, whether we'll ever go back to the way of life that we once considered normal. And especially during this time, we thought that it's actually quite important not to give up and stop everything. We wanted to interact with you. I personally actually really hate the phrase social distancing, because that's not what we are doing. We don't socially distance from each other, it's physical distancing. We're not in the same room, but we'll still talk to each other and interact, whether it's via video conferencing, Skype, Zoom, FaceTime, Messenger, and I have no idea what else there is. And for us, of course, for the Atom Society, this is our monthly talk where we normally interact, and this will now be delivered in this YouTube live stream format. So let me just check how many of you have joined. I now see 54 people connected to the live stream, which I think is great. I was actually not sure at all what I was expecting, and seeing so many of you connected and being interested is quite heartwarming for us. You will have to stay with us. We don't quite know how well this will work. We are all amateurs. We'll try to figure out how this works. You may have technical difficulties with this, which we obviously now can't help with. Actually, if you're listening to me now, you probably don't have technical difficulties. But anyhow, so tonight the format will be a little bit different than we'll normally do. We'll have to drop, at least on this first occasion, we will drop the science highlights that you normally send around and that we talk about, but we'll directly move to our speaker. We will actually have a break around 8 o'clock to give you an opportunity, if you want, to do the weekly clap for the NHS and thank them for the incredible work they are doing in these difficult times, despite the level of funding they received over the last few years. After this roughly 10-minute break, we'll continue. And at the end, as is traditional for a talk, you will have the opportunity to ask questions. Now, you can't ask the questions talking to us, but if you're watching this on YouTube and you are logged in, you should have a chat or a question box. And I've already seen one person, Neil Cuthbert. He told us he's here, you can see that. And so in the end, if you have questions, you can can type them into this box, and then Danielle, our speaker tonight, will try to answer them in the chat box. So there was something else I wanted to say. It's not quite clear. We may also have Joss Dorrington, which is the speaker of next month, joining us a little bit later. He had some private commitments, so it wasn't entirely clear whether he could join into the call. And if he does, then it will give you a teaser at the end of what next month's talk will be. So, and as a last comment before we really go live with the talk is, we want to get feedback from you how this really worked. Of course, it's not quite a replacement for an in-person meeting and the social interaction we all have together, but what you think about the talk, what worked, what didn't work, just let us know. Email the society with your thoughts. So that's enough of me today, and let's move to the talk. And I'm very pleased, and let's slightly change the format here. I'm moving this and getting rid of this. So I'm very pleased to welcome Danielle Fairbrass tonight. She got a Bachelor of Science in Sheffield in Biochemistry in 2011, and then a Master's in Biological and Bioprocess Engineering in 2012. And then she did a PhD, again with Sheffield University and Fujifilms, but actually I don't think that it had anything to do with photography. But then later, after she got her PhD, she went to France as a postdoc. And as far as I understand, she played with fast microscopy at that time. That was until 2018. And after that, she came back to the UK. She now does cellular engineering at Biomedica and really tries to support future gene therapies. And Danielle also joined the Atom Society last year as a committee member. And we have the pleasure tonight welcoming her to talk about From Pig Insulins to Gene Therapy, 100 Years of Biopharma. And you are already unmuted. Hi, Danielle.
Thank you very much, Alphonse, and good evening to everyone. Thank you for joining. I can see now we're almost at 60 people watching, so that's really exciting. Just one point of correction. I work for Oxford Biomedica, not Oxford Biotica.
Okay, sorry.
I'm not very good with robots, much more on the biological side of things. But otherwise, thank you very much for the introduction. I have got my alarm set for 7:55, so I will be able to let you all know when it's time to go and pack for our carers. So don't worry, we won't miss it, and I can resume after that. So if everyone's ready, I'm quite happy to start and talk to you today about, from pig insulin to gene therapy, 100 years of biopharma.
Okay, thanks a lot. I will just disappear until you're done.
Okay, perfect. Okay, so for pig insulin to gene therapy, 100 years of biopharma. Let me start by telling you a little story. Cast your minds back and imagine that you were living in the late 1910s, early 1920s, and your child has just been diagnosed with type 1 diabetes. Now, back then, the only treatment for diabetes was essentially a starvation diet to prevent the glucose levels in the body spiking too high. And unfortunately, what that meant was that for children diagnosed with type 1 diabetes, the patient lived for less than a year post-diagnosis, and for adults, less than 2. And so it really wasn't great. But then in 1922, 14-year-old Leonard Thompson was treated with insulin to treat his diabetes. Now, this was the first time this has ever happened. And as I said, prior to this, patients were expected to live less than a year, but Leonard lived for another 13 years and eventually died from complications unrelated to his diabetes. But his salvation was a culmination of decades of research and discovery, which ultimately led to the Nobel Prize in Physiology or Medicine in 1923 being awarded to Banting and McLeod for the discovery of insulin, although I should say they shared their prize with Best and Collett, their colleagues. But what was really important about this was that we now knew that we could take a protein that had been created by an animal and insert it into a human to treat a disease. So Eli Lilly, a massive pharmaceutical company, collaborated with the researchers that had discovered insulin to refine the extraction process, this time from pigs. The reason being that the meatpacking industry in the States is huge, and pork is an incredibly popular meat product, but the pancreas is a waste product. So they were able to get lots and lots of porcine pancreases cheaply in order to extract this insulin. And in 1923, Elatin, which is insulin created by Eli Lilly, it's just the trade name, became commercially available. And the animal-derived insulin was the primary treatment for type 1 diabetes for the next 40 years. Now, this has widely been accepted as the birth of biopharma. The treatment with insulin is known as the first ever biopharmaceutical treatment in the world. But you might be thinking, okay, but humans have been using animals, plants, fungi, and whatever, and their products to treat illnesses for thousands of years, sometimes to more success than others. So what makes this different? And the crucial step that defines the biopharmaceutical era is the use of technology to exploit these products commercially and to be able to engineer and design and create these products ourselves, rather than relying on nature to do it for us. So if we're thinking about this from a technological point of view, if we have the therapeutic protein that's our product that we want to make, what do we need to make this product? Well, first we need to know where to find the blueprint, right? If someone had made this product before, we would go to them and ask them how. In this case, we need to know how the cells know how to make this protein so that it is functional and it does what we need it to do. The next step is we need to be able to translate this blueprint. So if you were building a house, for example, and the architecture drew up the blueprints, but then the builders couldn't read them, then you're not going to get a house. So we need to be able to interpret this blueprint. We need to be able to package up these instructions in order to be able to deliver them in a way that is accessible and that doesn't lose any of this information. And we need to be able to deliver those instructions to where we're going to produce the product, which is our factory. So we also need to decide what kind of factory we want to have, what kind of factory is most appropriate. Now, this might seem a little bit conceptual, but hopefully you'll see where I'm going with this story as we travel through today. So early biopharmaceutical products such as insulin or, for example, human growth hormone were directly derived from animals or humans, with physicians relying on waste from the meat industry, as I said, or even cadavers for their supply, which isn't really what we're talking about with this engineering strategy. But then a whole heap of stuff happened between the 1940s and the 1970s that led to what we call what we would think of as modern biotechnology. And I'm going to take you through some of these really important steps this evening. The first thing I want to touch on is the discovery of the DNA structure. Now, I'm sure many of you are already aware of the story of Watson and Crick and Rosalind Franklin and how they determined this double helix structure of DNA. But the important bits that I want you to take away today are firstly that we now knew that this relatively simple structure of a sugar phosphate backbone surrounding these DNA nucleotides, which are adenine, thymine, guanine, and cytosine that you can see on the screen there, we knew that they were a code. They were a code that was incredibly specific, that if we could interpret it, could tell us how to make any protein that is made by any animal or plant. And the other thing that we learned was that they were complementary. So adenines will only ever pair with thymines, guanines will only ever pair with cytosines. And those are two really, really important things that came out of the discoveries in the mid-1950s. We then moved on to restriction enzymes. So what's a restriction enzyme? So restriction enzymes or endonucleases are a class of enzyme or a class of protein, in fact, that are able to cleave or cut up double-stranded DNA in a highly specific manner. And when I say highly specific, we now know that it's literally a short 6 or 7 sequence of nucleotides that the protein is able to recognize, and it will only recognize that sequence, that very, very specific sequence, and it will cut the DNA at that point. But they were actually discovered in labs researching lambda-phage. And lambda-phage is a hugely famous virus in the bacterial world that infects bacteria. And the way that it works is it infects the bacteria, let's say an E. coli, it multiplies inside the E. coli, and then as soon as it's reached the capacity it wants to reach, it will kill the cell so that it can be released into the environment. Now, what researchers noticed was that if they grew up some lambda-phage in a specific strain of E. coli and then tried to infect a different strain of E. coli with it, they got a much lower yield of lambda-phage at the end of it. And what they referred to was this restriction of phage growth, and that's why we call them restriction enzymes. And eventually, Arber and Nieselson discovered that this was caused by an enzyme cleaving the DNA that was contained within the phage, so the phage DNA, meaning that it was unable to replicate. And that's really important, and I'll point out to you why later. And the final thing that happened in the 1970s that was really key was that Frederick Sanger, a really quite famous British chemist, published the chain termination method for sequencing DNA. Now, this is quite an elegant protocol, actually, and it's really cool. What we knew was the protein that synthesized DNA. It was called DNA polymerase, and we knew how it worked. So we knew that it essentially looked at what we call a template DNA. It separated out into single strands and it photocopied it. So what Sanger knew was that if he put the bit of DNA that he wanted to sequence, which would be his template DNA, along with this protein and along with an abundance of these 4 nucleotides that make up the code, the A, C, T, and G that we have, into a tube, that the DNA polymerase would photocopy his bit of DNA over and over again. What he also knew was that if any of the nucleotides was slightly altered and the polymerase took one of the altered versions up, the reaction would stop. So what he did was he separated out into 4 reactions. He put his template DNA, so the sequence he wanted, the DNA he wanted to sequence, and his protein with an abundance of all 4 nucleotides into 4 separate tubes. Then in each tube, he put 1 nucleotide that was slightly altered. So for example, here in the green tube, we've got a slightly altered cytosine. So what that means is you end up with a whole host of different strands of different lengths, because the DNA polymerase will take up this altered nucleotide at a different point. It's completely random. You're then able to separate these pieces out by size using a process called gel electrophoresis that I won't go into. But ultimately, that means you can read down the gel as if you're reading a piece of text, and you can read the sequence. So here we would have reading down A, G, A, C, T, C, and so forth. And the reason I say that this is really cool is not just because he won the Nobel Prize in 1980 for chemistry for it, but also because we now have much more sophisticated methods of DNA sequencing that mean we can sequence much more DNA much more quickly. But whenever a new method comes out, it is the chain termination method that Sanger developed in the 1970s that we use to validate this new technique. And that's why I think it's super cool. So if we go back to our key things that go into engineering and making a new product, then we now know where to find the blueprint. Watson Crick published where to find it. It's in the DNA. We also know how we can manipulate that DNA so that we can select only the bits that we want to make only the protein that we want. And more importantly, we now know how to figure out which bit of that DNA we want, which bit is the sequence that we want for the protein, because we can now sequence it, which means we can now read it. So while all this was going on in the molecular biology labs and the chemistry labs, a whole heap of stuff was going on in tissue culture labs as well that were really exciting. And I really recommend that you go and have a look at this book chapter that I popped on the bottom there. But I'm not going to focus on all of this. I'm mostly going to focus on what happened from the 1950s onwards, with a minor point that the ECAC that you've got up there in 1920, that was established to preserve cell cultures, and it's still going today. It's now essentially a massive biobank that researchers can contact to request any cell line that they want to use. But the important thing that happened for our story happened in the 1950s, and that was when we figured out what immortal cell lines were. So immortal cell lines were defined by Hayflick as a life form capable of indefinite survival in conditions where no changes have occurred in molecular composition from some arbitrary beginning, which frankly doesn't really clear it up. But most cell types in the body, as we know, do not proliferate indefinitely. This is essentially why organisms age and die, because most of the cells in your body have a lifespan. They can't proliferate indefinitely. But what we did notice was cells that were extracted and cultured in vitro, so in dishes or in flasks, exhibited immortal behavior. And we noticed this throughout the 1940s through to the 1960s. We now know that immortal behavior can occur spontaneously. This often happens in murine cell cultures and generally rodent cell cultures, which is why a lot of the more famous ones maybe have come from hamsters and mice and so forth. But we also know that it can be induced. The reason we know it can be induced, especially in human cells, is because of our observations of cancer cells. Cancer cells are genetically changed and altered to the other cells in your body to be able to cope with the stresses of indefinite proliferation. That's why they're a problem when they're in your body, because they don't stop multiplying. And what we knew was that if we used cancer-causing viruses or chemicals or even radiation, we could induce this immortal behavior in cell cultures that we had extracted from human bodies. And it's at this point that I want to touch upon the importance of Henrietta Lacks. I'm sure a lot of you have heard of Henrietta Lacks or maybe heard of HeLa cells. But essentially what happened was this young woman was diagnosed with an aggressive cervical cancer in 1951, and her cancer doctor took her biopsy and sent it to the director of the tissue culture laboratory at the hospital that she was at. Well, people working in the lab noticed that they could culture her cells indefinitely, at first in chicken plasma, and then eventually they noticed they could do this in pretty much any cell culture media that they decided to try it with. And this was huge. These cells were shipped to laboratories in academic institutions and pharmaceutical companies worldwide, predominantly so that people could use them to study cancer. But what also happened was it meant that it allowed researchers to discover how they could immortalize cells, and this could be done for the purposes of protein production. I should highlight that all of this was done without the permission of Henrietta Lacks or her family, which at the time was legally permissible. But this continued so much that in March 2013, researchers published the genetic sequence of a strain of HeLa cells, again, without seeking permission from Henrietta Lacks' family. An agreement was later reached that year that means they regained some control, but it started an entire conversation about ownership of genetic sequences and whether a patient can own their DNA sequence after it's been removed, after the cells have been removed from the body. And it's a conversation that is actually still ongoing in some parts of the world, although Europe has legislated to address it. So we now know what kind of factory we want to have. We want to have these immortal cell lines that we can manipulate genetically to get them to make the protein that we want to make. In 1948, the first immortalized cell line was established. So the first cell line where researchers immortalized it themselves. Crick and Watson then told us where the blueprint was. Arber, Nathans, and Smith told us how to manipulate DNA, how to play around with it. And then Frederick Sanger told us how to read it. And so the combination of all of these things meant that in 1986, the first commercial therapeutic was produced rather than just harvested. It was produced using Chinese hamster ovary cells. So I told you that a lot of cell lines are derived from hamsters. This was a cell line that was established in 1957. And we also know that now, today, 70% of recombinant protein therapeutics, so 70% of these commercial proteins are produced in CHO cells. So it's still a cell line that is widely used today. The vast majority of these recombinant protein therapies are monoclonal antibodies. What are they? So you probably heard of antibodies. They're a component of our immune system that are able to recognize what's known as antigens. So antigens are normally proteins or molecules that are presented by a pathogen that's in the body. And the antibody will bind to these antigens and then alert the immune system so that the other immune cells can come and kill the pathogen. Monoclonal antibodies are antibodies simply that are derived from one clone or one cell. And what we do in terms of generating them therapeutically is we would isolate that one cell that makes the antibodies and allow it to proliferate. What's really important about that is it means that all of these antibodies come from a genetically identical source, which is really important when we're talking about designing the genetic sequence to design this protein. And the other important thing is, as I said, they're highly specific and mean that you can tailor therapy to a designated target. When we think about cancer, for example, chemotherapy involves flooding the body with toxic chemicals and just hoping that the cancer cells take them up quicker than the rest of the body, which means you get really bad, wide-ranging side effects. And the hope is that with antibodies, you can drastically reduce this. So monoclonal antibodies were originally produced, using the hybridoma technique. So we talked about using cell lines as our factory, but at first, the factory was the mouse. This technique was developed in the 1970s, and it is still used to generate antibodies used in biological research and diagnostic techniques. And essentially what you would do is you would immunize the mouse with your antigen of choice. So you would just stick this antigen into the mouse's system, and it would then produce antibodies against it. You then take out all of the antibody-forming cells from the mouse and fuse them to tumor cells to form hybridomas, essentially immortalizing these antibody-forming cells. You would then screen them to isolate the single cells that are producing the antibody you want, because there'll be lots of them in there, and then you can extract your antibodies from that. Now, the reason we don't really use this anymore to make therapeutic proteins is partly because the process is slow and difficult to scale up, which makes it incredibly expensive, but also because proteins generated using mice or indeed any animal will trigger an immune response in the body. And the antibody that you're putting in to treat a disease will be treated by the body as an antigen. And the patient will produce anti-drug antibodies, which is really not what we want. So improvements in protein design basically meant that we were able to move away from murine antibodies all the way through to be able to, using restriction enzymes to design our genetic sequence, to be able to generate human antibodies using cell lines. And that's where we're at now. And I will take this moment to pause because it's just coming up to 8 o'clock. So I'll let you all head off to clap for the carers, and I will see you at 8:05.
Hi, everybody. I'm just back from outside. We even had a bride clapping on one of our neighbors in her wedding dress. Not sure if she was actually a bride. Anyhow, I think it's almost time. Let me see. Yes, Danielle is there as well. And I think we can give it another 30 seconds or so. And then it's all to you again.
Shall we?
Yep, go ahead.
Okay, so welcome back, everybody. So I was just explaining to you how researchers had improved protein design through genetic manipulation using these restriction enzymes to get to a point where we can use cell lines, so for example, this tiny cancer ovary cell line, to create human antibodies, so using a fully human genetic sequence rather than using a murine sequence, and hopefully that can reduce the immunogenic response. So what this means is, in terms of therapeutic proteins and monoclonal antibody protein production specifically, is that we now have tailored, highly specific therapies that are able to treat a wide range of diseases. And so just some of the, and I hope that this is not at all an exhaustive list, but just some of the diseases that we're able to treat with monoclonal antibodies include autoimmune diseases such as rheumatoid arthritis, Crohn's disease, and ulcerative colitis, as well as moderate to severe forms of asthma, right through to HIV or wet age-related macular degeneration to prevent people from going blind. And that's before we even talk about the roughly 30, just over 30 monoclonal antibody therapies that have been approved for the treatment of various cancers, which is the type of disease that this particular therapy has really found its niche for. And these include non-Hodgkin's lymphoma, breast cancer, and colorectal cancer. So we were talking before about this factory idea and about how at first monoclonal antibodies were made in mice, but then now we use cell lines. So how exactly do we do that? And so we now have this genetic sequence that we can read, that we can cut up, and we can load it onto our vehicle. And in the biological world, we refer to this as a vector. Now, I know those mathematicians out there will be thinking that vector is something completely different, but in this case, a vector just means a vehicle to carry genetic information. And so we load up, in this case, our circular DNA vector, and we transfect our cells with it. Transfection is a process that can be done in many different ways, but it essentially is just a process that safely inserts DNA into cells without killing the cells, and can be done using chemicals or using electricity to poke little holes in the cell membrane, for example. We also insert what's known as a resistance selection marker. So basically what we want to make sure is only the cells that have had this vector come into them and have taken up this genetic information are the ones that we're dealing with. So we also introduce to them on this vector resistance, usually to an antibiotic. And once we've done the transfection process, we expose our cells to this antibiotic, and any cell that isn't carrying the information for the therapeutic protein that we want to make dies. We then take this pool of cells that's producing our therapeutic protein, and we dilute it out and plate it out into 96 well plates, or even a higher number of well plates, at such a dilution that statistically 0.5 cells are in a well. And what this means is in some wells there'll be no cells, and in some wells there should only be one. And this is to ensure that monoclonality that we talked about before, that we only want to have one cell in there to make sure that all of the cells that come from it are genetically identical. We expand up these clones, we select out our best producers, and we cryopreserve them so we don't lose all of that work. We then do a second round of clone generation using these clones. So we plate them out again, we expand them up again. This is a regulatory requirement, and this is basically because if you are a researcher staring down a microscope at dozens of 96-well plates, you can make a mistake. So you might think that you've looked at a well and only seen one cell, when in fact over off in the corner on the edge of the well, there was another one. So this is just essentially to statistically ensure that if you're saying that it's a clonal cell line, it is a clonal cell line. And the more rounds that you do, the more sure you can be, but obviously it takes a long time. So for regulatory requirements, you're only required to do 2. You then again cryopreserve the clone that you selected as your high producing cell line, and you culture this cell line in a stability study, which is essentially where we do this continuous culturing, this continuous proliferation, for usually around 12 weeks. What we're doing here is we're ensuring that this DNA information, this genetic information that had been inserted into the cell, has been stably integrated into the cell's genome. So any DNA that's floating around in the cell will be read, and the protein that it encodes will be made. But as the cells divide, you lose that information over time because it's just in the jelly that's inside the cell. It's not in the genome, so it's not replicated. What we want to check is that our DNA has stably integrated into the genome, so that every time a cell divides, it takes that information with it into both generations. And we do this for about 12 weeks because it normally takes about 4 weeks of continuous culture to get from your normal lab scale 20 milliliter culture up to the 2,000 liter bioreactors that we use for manufacture. And you want to make sure that the cells are still capable of producing your protein at that point. Now, this is a really long and laborious process. But luckily, some fun new gadgets have come onto the market over the years to improve this, to make it faster, to make it more accurate. One of those is the single cell printer, which was developed by Cytina and has come out quite recently, actually. And it uses a combination of a little camera and a vacuum pump. And it goes over the wells in your plate. And the vacuum pump is pulling everything away until it sees that there's only one cell in its little nozzle, and then it will turn off the vacuum pump, the cell will drop into the well, and it will move on to the next one. So whereas before we were averaging half a cell per well, so only half of our wells would probably have clonal cells, and then we now know that all of our wells have the potential to become a clonal cell line. So you're already doubling the number of clones that you can screen at once. And then you combine that with something like the NI1 compact image cytometry machine, which is essentially a really sophisticated camera that can look into the wells of your plate, and it can say, oh, there's a cell in that well, and it'll highlight it with a little blue circle, as you see there. And then it will track the growth of that cell over time as you keep imaging it day after day after day, so you can see how your colonies are growing. And for both of these, you have photographic evidence that there is only one cell in that entire well. And then you've got things like the ClonePix mammalian colony picker, which does some very similar things to both of these things combined, along with some other stuff. And it's that this one specifically is used quite often in monoclonal antibody production. And you get images like the one here on the bottom right. The green images here are showing where there is cell growth within that well. And so you can see which colonies are doing better than others, for example. Now, what's really cool about these gadgets is that if we combine, let's say, the use of the single cell printer to accurately know that we're having one cell per well, doubling the clonal colonies per plate, and if we combine that with imaging using the NI1, what the regulators have said is that because there is photographic evidence tracking the monoclonality of these cultures, you can completely eliminate your second round of clone generation. You don't have to do another round because you've proven that your cell line is clonal. This reduces your development time by up to 9 weeks, and you've already been able to screen double the number of clones. This massively improves your chance of getting a really high-producing cell line that can make your therapy of choice. So this is part of what I do on my day-to-day, and I think this is really cool. So what next? I've spoken to you about recombinant therapeutic proteins, so therapeutic proteins that we're making by combining genes that we want to use onto a DNA vector. And as of 2017, over 140 recombinant therapeutic proteins were approved for use, the vast majority of which were monoclonal antibodies, and the market size was about 91 billion US dollars 3 years ago. And advances in our genetic manipulation techniques and cell line development have made these highly selective therapies a reality for thousands of people, with production yields 100 to 1,000-fold those seen in 1986, which means we can treat more people for less money. So why haven't we cured all diseases if these things are so fantastic and now so theoretically easy to make? I mean, the first thing to say is that not all diseases have a specific antigen to raise an antibody against. And another way of saying that is that not all diseases are caused by a single protein or can be cured by eliminating a single protein. And the other thing is the delivery of the genetic material. So far we've talked about using these vectors to get the genetic information into cells, to get them to produce the protein, and then we insert the protein into the patient. But what if that isn't possible? And what we actually want to do is to deliver genetic information directly into the patient. We can't do that by transfection, this method that I talked to you about earlier. We can't flood the body with electricity to poke pores in the cells in your body because, I mean, ultimately you would die. It's not feasible. So that brings us on to cell and gene therapy, and that's what I want to talk to you finally in this last section of the talk today. Cell and gene therapy are overlapping fields of biomedical and biopharmaceutical research and refer to the use of cells or genes to treat disease. So we're not just using proteins now, we're using cells or genes. So in cell therapy, we remove cells from the patient's body and keep them alive in a sterile environment, something which we've known how to do since 1907. And they are then treated to encourage expression of an absent protein or to encourage expression of a therapeutic protein, much in the same way that we were treating our cell lines before to get them to make our monoclonal antibodies. The cells are then inserted back into the patient's body where they will proliferate and they're able to treat the disease. In gene therapy, we take it one step further, and the genes encoding the therapeutic protein are directly inserted into the patient's body via a vector or a vehicle. And these vectors are usually targeted to the affected cell type because we don't necessarily want to insert this genetic information all over the body. If you want to generate a protein that just needs to be made in the liver, you don't want it being made all over the body. You're going to have what we call off-target events that are going to cause immunological reactions. So up to now, we've talked about the blueprint, we've talked about how to translate that blueprint, we've talked about how to load it up, and we've talked about the type of factory that we want. And the key for this talk, this part of the talk, is talking about this transport of instructions, these vectors that we mentioned before. I want to focus first on gene therapy. The vector that we use for gene therapy, the vehicle that we use to insert our genes into the patient's body, are viruses. Viruses have evolved over millennia to effectively insert genetic information into host cells. It's exactly what they do. It's the only way that they're able to replicate. And they were proposed as a mechanism for gene therapy as early as the 1980s. They're already regularly used in research labs to carry genetic information into cells or into entire animals, and so it's a technique that we know how to do. But considerations for developing this for therapy are that the therapy needs to be specific, so it needs to only target the cells that we want to treat, and it needs to be safe. And that's twofold with viruses. Not only does it need to be safe for the patient themselves, it needs to not be able to replicate and go on to infect other people or animals or whoever. So there are two classes of virus that are used in the development of gene therapy. The first is the adeno-associated virus, or AAV, and the other is the lentivirus, LV. So AAV-mediated gene therapy is one of the more extensively studied. AAVs are very simple viruses. It's just a protein shell surrounding and protecting the DNA and genome inside. And it relies on the assistance of other viruses for replication. So this is really important for our safety considerations, because if we're only infecting the patient with AAV and not with its assistant virus, it can't replicate. So it's perfectly safe. The DNA delivered, however, doesn't integrate into the host cell genome. So where we were talking before about having this stable integration, so you're going to have continuous production of this therapeutic protein, this cannot happen with AAV. So it will be diluted over time, making AAV-mediated gene therapy more appropriate for cells that don't have a high turnover rate, for example, in the eye. Finally, AAV have been shown to be less immunogenic than other viruses, which is good. But because of the nature of the virus, there's a lot of them around. And so a lot of people have already been exposed to some form of AAV, which might mean that the body already has an immune response to that virus, which potentially would make the therapy unusable. There has been an AAV-mediated gene therapy approved for therapeutic use. It was approved a few years ago, called Luxturna. So Luxturna is a treatment for Leber congenital amaurosis, which is a rare inherited eye disease that appears within the first few months of life. About 18 different genes are associated with this disease, meaning there are about 18 different variants of this disease. But for all of them, the current standard of care is supportive. You basically have to teach this child how to live as a blind person because you cannot restore their sight. Luxturna is able to do that for one of these 18 cases. So it carries a gene for RPE65, which causes about 6% of cases of Leber congenital amaurosis. And data as of last year, this is how new this therapy is, show that improvements are maintained up to 4 years after treatment. So these kids have been given their sight back, or at least partial sight back, and that has been maintained for at least 4 years and may well be maintained further. We just don't have the data yet. Lentivirus-mediated gene therapy. So lentiviruses are named because in their wild-type version, after the initial infection, there's a latent period. So there's a long period before disease progression. And examples of lentiviruses include HIV, which I'm sure you've heard of, SIV, which is the simian or monkey version of HIV, and EAIV, which is essentially equine or horse version of HIV. The current lentivirus vectors for gene therapy, as they stand, are the product of decades of engineering to prevent wild-type virus particles forming and replicating. I'm sure as soon as you saw HIV there, you got a bit worried. We don't want to be infecting patients with wild-type HIV. And indeed, in the first few years of lentivirus-mediated gene therapy, there were a lot of issues because these vectors were just not safe. But we're now at a point where lentiviral vectors for gene therapy are incapable of forming wild-type virus particles, and that's super important. What they are capable of doing is infecting a wide variety of cell types, depending on which form of LV you choose, and they do integrate their DNA into the host genome. So they can be used without the loss of the therapeutic gene over time, which means they can be used for replicating cell types, such as epithelial cells. So one of the targets for LG gene therapy at the moment is cystic fibrosis, to replace the membrane protein that's mutated in cystic fibrosis, which would not be possible with AAV. LV can also carry about 3 times more DNA than AAV, which is hugely important because, for example, for membrane crossing proteins that are an issue in cystic fibrosis, they're way too big. There's too much information that needs to be carried for AAV to do it. So there aren't currently any LV-mediated gene therapy products on the market, but one that is showing real promise and is in later stages of clinical trials is prosavin or axolenti-PD. And this is a treatment for Parkinson's disease. So we had a really interesting talk a few months back that I'm sure a lot of you remember about Parkinson's disease, and I know it's something that's close to a lot of your hearts. And the current standard of care is oral L-DOPA. And the problem with oral L-DOPA is it becomes less efficacious over time. The longer the patient is taking it, the less impact it has on their symptoms. Prosavin was an LV-mediated gene therapy based on an equine virus that carries the genes for all 3 enzymes involved in dopamine production. So rather than just trying to prop up the production of dopamine by increasing the amount of L-DOPA, we're restoring the brain's ability to make dopamine for the entire chain of production. Early clinical trials for prosavin showed significant improvements in motor behavior a year after 1 dose, and continued moderate improvements above basal levels up to 5 years after treatment. And I repeat, that's just after 1 dose. They're not having to take L-DOPA. Extensive genetic modifications to the design of the genetic information that the virus was carrying led to the production of what's known as axolenti-PD, which is essentially an updated version of prosavin. This hasn't gone into clinical trials yet, it's just about to, but animal and preclinical trials indicate that it could have up to a 10-fold increase in dopamine production in comparison to prosavin, which means that you could inject the patient with less and they'd still have as good a reaction. The final thing I want to talk to you about today is cell therapy. So the best example of cell therapy that I can talk to you about is something called CAR T therapy. This is where T cells which are a type of immune cell, are removed from the patient. And using this viral vector technology to deliver genetic information to these T cells, they're modified to be able to recognize cancer cells. These modified cells are then reintroduced to the patient, they replicate, they find the cancer, and they're able to kill it. And in theory, the patient is cured. In 2017, Kymriah was approved by the FDA for therapeutic use. And this is a CAR T therapy that is used to treat a childhood leukemia that ordinarily would lead to death within a couple of years. But the first patient that was treated with it in the initial trials, she was treated with it when she was 9, and she's now in her late teens. And she's had no remission. This is a product that's made by Novartis, and the viral vector that's used to infect these cells is made by Biomedica, which is the company that I work for, and a company obviously here in Oxford. And what's really exciting about Kymriah, some of you might have heard the name before, is that in 2018, NICE struck a deal with Novartis that meant that this could be offered on the NHS. Patients in the UK do not necessarily have to have this treatment privately because it is now available in the NHS. And last year, the first child in the UK was given this treatment on the NHS. And I think that's just something that's incredibly fantastic and incredibly exciting for the future of medicine. So in summary, almost 100 years ago, I was a little bit cheeky with my title because it was 1922, not 1920, but almost 100 years ago, insulin was administered to Leonard Thompson in what would become the first ever biopharmaceutical treatment. And since then, advances in knowledge about DNA, its structure, how it works, and how we can manipulate it intra and extracellularly led to the birth of modern biotechnology and enabled scientists and physicians to engineer biology to design complex and highly specific therapeutics. This has led to a multi-billion-dollar industry that's dominated by monoclonal antibodies and means that we can treat diseases ranging from transplant rejection to immunodeficiency, to cancer, and to a whole host of others. But that's not enough, and further advances in genetic engineering techniques combined with improved safety profiles of the virus vectors that we want to use have opened the door to safe and effective cell and gene therapy. And these have the potential to cure life-altering conditions, not just treat the symptoms. And that's the exciting next chapter of the biopharmaceutical industry. And with that, I would like to thank you for listening. I can see there are already a couple of questions, and I welcome any more.
Okay, so thanks a lot, Danielle. That was really interesting. I think I'm working in the wrong area. Yes, so you see the question. So maybe we start with the first one, which is of course quite topical. Could you say how different vaccines for COVID-19 would work, I assume, or how they will be produced? Is that related to the work you're doing?
So there are some. So there are a whole host of potential vaccines that are being developed all over the world at the moment. And there is actually an article from the scientist that I can share on the Atom Facebook page later that summarizes the ones that are currently in clinical trials or are expected to be in clinical trials later this year. And several of them, including one that's being developed in the UK by Oxford University, and that Oxford Biomedica are actually involved in scaling up manufacture for, is using an AAV-based vaccine that was developed for monkeys, combined with technology for a HIV lentivirus, a HIV vaccine for, again, AAV-based. It's combining these two things to try and target the envelope proteins. So I didn't really go into the structure of viruses, but on the surface of the protein shell, it's made up by what we call envelope proteins. And so this AAV-based vaccine would enable immune cells to target these proteins on this virus. And there's a couple of different vaccines that are in clinical trials that are based on AAV or AAV-like treatments. There are others that are based using different technology. And if you, I will share that the article from the scientist, because it describes the type of therapy that they're all involved in.
Okay, I think as we are in this area, Andy had an interesting question. As COVID is so efficient in infecting cells, can that actually have in the future a therapeutic use if you make like a safe version of this coronavirus?
So I should highlight that I'm not a virologist. My area of expertise is not in viruses. What I would say is that the key thing is making it safe. From what I understand, SARS, which is the type of virus that COVID-19 is not a lentivirus, and it behaves in a completely different manner. And I don't know whether we'd be able to engineer safe technology. And you've got to consider as well that we started engineering lentiviruses back in the 1980s, and we're only at a point now where they're safe enough for us to use in therapies.
Yeah, so, just barely 40 years.
Sorry.
So barely 40 years later.
Yeah. And the other issue is specificity as well. We want to make sure that we can target these viruses to different cell types. And that's what's really cool about LV, is that we can use, there are so many different types, there's so many different proteins that we can target them to specific cell types. And that's not necessarily something that's going to be possible with a coronavirus type virus.
Okay, here we have the next question from Jane. In the stable cell line generation, you said you use antibiotic resistance as a marker. So are your cell factory bacteria, and if so, which ones?
Yeah, that's a good question, and I should have clarified at the time. So no, the cell factories are mammalian cells, usually. Sometimes they're insect-based cells, but often for therapeutics, they're mammalian cells. Actually, mammalian cells and animal cells are also susceptible to some types of antibiotic. In high concentrations, if you took the antibiotics that you took from the doctor in really high concentrations, they would make you sick. So it's just that we're able to use ones that we know specifically inhibit the growth of mammalian cells at a really high concentration, because it's not in a human body, so we're not going to kill someone. Okay.
Another one here from Bob. In the factory production line, if the stable cell line process fails after this 12-week study, then presumably you have to start again from the beginning of the development process?
So not entirely from the beginning. As you might have noticed that at certain points I said that we cryopreserve some of the clones that we have generated. And the reason we cryopreserve some of them is because we've already checked that they produce the protein at a level that we're happy at least in the first instance. So normally by the time we get to the point of having a stability study, we would normally have at least 2 or 3 different candidates that we would want to test. And we might take all of these to stability study level, so then we can pick the best of those 3. Or we might only take 1, knowing that we've got backups. So normally that's only going to put us back about 12 weeks. We're definitely not going to have to go all the way back, because we check at every stage that we've still got cells that are producing.
So you just have to go 1 step back, effectively.
Yeah, so it's just 3 months rather than half a year or a year.
Bad enough.
Yeah.
So, and we have a question from you. Do you think, and that's really on the non-technical side, do you think that the cultural and religious complaints some make concerning the ethical boundaries of work like yours will ever go away? How can science work with complainants
to reduce the friction?
To reduce the friction, yes. With complainants, yes, okay.
Yeah. It's really difficult. So the fact that we're able to produce most of our therapeutic proteins now in cell lines drastically reduces this ethical constraint that people have. These cell lines were isolated back in the 1950s and 60s. We don't generally make new cell lines now for therapeutic production, because we already have ones that work perfectly well. The main ethical issue, I guess, is in terms of the clinical trials afterwards for us. That's not really something that we can get away from at this point. And I think the main thing that would improve this is if we were able to improve people's education about why we still need to use animal models to test our products, and the amount of work that goes into avoiding the use of animals at all costs and at all possibilities, and the amount of ethical boards that people have to satisfy before they're allowed to do animal testing.
Okay, and there's a question from Jens about cancer treatment. So what limits the CAR-T mechanism to certain types of cancer? Is there hope that more generic T-cell based cancer therapy may work, or can that work?
Yeah, so thank you for that question. It's really good. So the whole point of this is that it's highly specific. The only thing that limits the mechanism at the moment to certain types of cancer is the amount of research that's being done on it and the point at which we're at in terms of clinical trials and FDA approval. Its beauty is in its specificity, and you can't really have generic cancer therapy, because cancer is actually a word that describes thousands of different diseases. And that's why things like chemotherapy and radiotherapy will work for many different types of cancer, but also cause a lot of problems for the patient because they're generic treatments. We like the fact that this is specific, and we want that to continue. But yeah, it's a case of time and money in a lot of cases.
Yeah. Okay. So there is hope. And Victoria had a question. I don't think they are silly questions. Once you mentioned that any bacteria is much, much larger than a virus, why can't we use antibacterial soap against viruses?
So this is, again, a lack of education. Marketing companies from soap production marketing departments of soap production companies have managed to convince everyone that antibacterial soap is superior to normal soap in terms of combating virus infection. Any soap is good. Any soap will help break down that protein envelope that I was talking about earlier. There's also the friction of rubbing your hands together. The actual physical motion of doing it helps to break those down. And literally any soap will do that. Washing up liquid will do that. Shampoo will do that. The only thing I would say is that liquid soap is possibly slightly superior to bars of soap, simply because if you don't rinse the bar of soap afterwards, you might end up with some particles laying latent on the soap. But that's very low risk. If that's all you've got, that's what you should use.
Okay. Okay, there are a few nice comments about this, and I think question-wise, we have covered everything. And before we close this live stream, we actually have Josh here. He's our next month's speaker. Let me try to add him to the stream. Thanks a lot, Danielle. I will remove you and give all the floor to Josh, just to tell us a little bit what he will be doing in a month from now.
Hello, I'm Josh. I'm a PhD student, University of Oxford. I study the weather and the predictability of the weather, trying to work out why sometimes we produce really great forecasts and sometimes we get absolute busts that you can't predict at all. And next month, I'm going to be talking about the history of weather forecasting. How did we go from wise men and women looking at sticks to determine the weather to the modern era of satellites and supercomputers, and how the question of weather relates to the climate. So yeah, join me for that.
Okay, thanks a lot, Josh. I really want to thank everybody, not just Danielle and Josh, for making this live stream a success. I think it was quite important and quite encouraging to see we had over 60 people attending, and I think the question and answer session via YouTube was more or less exactly like we have in the normal talk. So well done, everybody. I think, well, this will continue. But anyhow,
Yes.
Yes.
I just want to thank you to everyone for tuning in and listening. I'm really pleased that we started off with about 56 viewers and we still have about 56 viewers. So hopefully that means I wasn't too boring. It's a lot easier to walk away than it is when you're in person. You can't walk out the room then. And with my Atom secretary hat on, I would also like to reiterate, as Alphonse said earlier, if you do have any feedback, please do get in touch with me via the secretary email account. And what I might also do next week is send out a short, really tiny survey just to gather some sort of response about how you feel everything went and whether you think any improvements can be made. This is completely voluntary, obviously, but I'd really appreciate it if you could engage with that.
Okay, with these last words, thanks a lot, everybody, and this ends this live stream. Thank you.
Thank you.
Bye.
Bye.